Well, I'd like to thank the organizers for asking me to speak, and I had this sudden urge to download a photograph of John Cleese at the BBC News desk saying NAPR something completely different, but I couldn't find it quickly enough, so I left this lovely photograph of the skiffs on the Charles River in the first snowstorm in Boston last year in honor of the fair city that we are in fact in today. The reason I say it's going to be something completely different is I think I'm the first academic to get up here, except for William, who is pursuing his PhD. I assure you I'm pretty far out from pursuing my PhD, unfortunately. But because I'm going to talk, I think, more globally and about what might be considered a bit esoterica, and I have three ideas that I want to convey to you today. The first is a sort of a brief reflection on what we're all really worried about here, this notion of immunologic tolerance. And then hone in very quickly into a B-cell centric perspective, really focusing on, I think, the last 10 years in dealing with how we've appreciated that there's a degree of plasticity to B-cell tolerance that wasn't previously appreciated because of the discovery of a couple of new molecules. And then what I'd like to do, I'll show you a little new data that's, well, depending on how the reviewers at JEM feel, may or may not be appearing in print fairly soon. But we sent back all 64 panel figures now to them, so I think they'll be happy with it. At any rate, and then finally, I think talk a little bit about how using this particular system, we can dream about repertoire manipulation. And I think that's really going to be my message: is that we really need to be thinking about this as we move into the future. That it is possible now to actually tread where angels fear. Nowadays, and actually believe that we can change repertoires of immune receptors. And so I'd like to remind you that what we're really all talking about here today has to do with Peter Medawar and Macfarlane Burnet in the late 40s and early 50s, as well as Ray Owen at Caltech, where they came up with this notion of an immunological self, either through skin grafting experiments where they showed that isografts succeed and allografts fail, or in the dizygotic twinning experiments of Owen, where he showed that the genetically disparate individuals that shared placental circulation could accept grafts one another. That is, this idea that the concept of self and non-self is in fact acquired and is in fact not hardwired. This, of course, led to discarding the current paradigm of immunology back in the mid-50s and having this one be the one we now accept. And the clonal selection paradigm, variously attributed to Yerney or Burnet or Talmage, I think it's Burnet personally, could deal with all the old ideas that we had known existed for the immune system, but also could deal with this notion that by having differential fates, by having the immunologic specificities quantized, so each cell expresses a single variety of receptor and hence has a single effector function, you could you could purge the repertoire at varying points by either eliminating or functionally inactivating clones that show you didn't want responding, and only leaving those that you felt were safe. This doesn't turn off. And so to move right into the B-cell world, this more or less translated to, I'm going to use my green one because it will turn off. It will translate into these classical ideas that the BCR itself, through the signaling machinery, is delivering signals that affect these binary fates. And sure enough, it has become quite clear with time over the last 30 years from the work of hundreds of laboratories that indeed, once you do express an antigen receptor here in the immature bone marrow B-cell pool, the different receptors are depicted by different colors and shapes here, that you will either replace this receptor through a process known as receptor editing, or in fact eliminate the cells through deletional central tolerance, such that the emerging repertoire is purged mainly of self-reactivity, and then once cells enter this immature or transitional state in the periphery, which we and others described in the early 90s, that it undergoes an additional round of this deletional or even failed positive selection forms of elimination, so that the emerging mature repertoire is presumably screened pretty heavily for unwanted specificities. And this was more or less the paradigm, I think, up until about 2000. And at that point, this family of receptors and ligands burst on the scene, this TNF family member BLyS, or BAF, we should call it BAF, I guess, since we're so close to Biogen IDEC. It's right down the street. And it interacts with one of three receptors, TACI BCMA or BR3, and two of these, TACI and BR3 or BAFR, are in fact expressed from this immature stage onward, and it became pretty clear pretty early on that the interaction between this cytokine and this receptor was really key to the selection and homeostasis of everything from here forward in the B cell pool. Now, I don't want to give a talk on BAF, and I've put this Magritte up here to give you, to kind of make the point I want to make, which is I've got to tell you about it, and indeed we are talking about this, but I think there's more to the picture that I want to convey today than just this cytokine and receptor. So bear that in mind as we go on. So rather than show you all the data for what I'm about to tell you, I'm going to summarize it here. What's quite clear is that all B cells from here forward rely on this cytokine to stay alive, number one, and even more importantly that the stringency of this selection point is variable based upon how much BAF or BLyS is available, such that in contrast to the usual 2/3 reduction in cells moving from this stage to the mature pools, if you have an enormous amounts of this cytokine, virtually everything, good, bad, or indifferent, will move forward. And it's only when the system is replete with essentially BAF utilizing cells, as depicted here, where you have light purple, so there's not too much available because most of it's bound to these cells. These guys begin competing with one another for this cytokine, and only those that are fittest, that is, that have a BCR delivering a moderate tonic signal, move forward into the mature pools. And this raised the idea that this last tolerogenic step is in fact plastic, and that one might want to both worry about it and possibly explore it. Now, what's occurred to us, and this is a prelude to the sort of newer data I'm going to show you, is that in addition to sort of circumscribing the biological space for the pre-immune follicular and marginal zone pools, is it possible this cytokine receptor family also plays a role in subsequent selection steps? I'd like to focus your attention right here. This is something that only B cells do. I mean, I shouldn't say that. T cells do in the germinal center, and T follicular helpers are, of course, absolutely critical for the formation of germinal centers. But in the germinal center reaction, B cells undergo some pretty interesting changes that also means there's going to be another selection step that has to be dealt with. And just to remind you what germinal centers look like, they are these. This is at 14 days post-immunization with a T-dependent antigen. And what you see is that in the, this is the periarterial lymphocytic sheath, or the T cell zone in the splenic white pulp. These are the follicles that are the mature pre-immune or naive B cells. And the GCs are these little red structures stained with peanut agglutinin that if you then focus in on this one, which is anatomically correct because of the plane of the section, it's divided into what were classically determined by H&E staining, the light zone and dark zone. And what you can see is that in the light zone, this is where the majority of the T cells standing with TCR beta are found. The dark zone is mostly dividing B lymphocytes. And they always form at the T-B cell junction. But the key for today's talk is that in this unique reaction, B cells undergo AID-mediated point mutations, and through some miraculous selection process that we know involves TFHs, follicular dendritic cells, that are very specialized cells found in the GC, interacting with B cells, that as this mutational process goes forward, cells that reacquire autoreactivity, and that's going to happen because we are mutating in the V regions, will in fact be selected against. Cells that lose reactivity or retain only low affinity for the immunizing antigen are also selected against, and only higher affinity B cells make it out of here into the memory and long-lived plasma cell compartments. Now, this is a big deal because it means that not only do we have to winnow the repertoire of B cells at these developmental checkpoints that we talked about before, such as the immature bone marrow or the transitional peripheral stages, you have to do it again in the germinal center. And what's always defied explanation, I shouldn't say defied, but what none of us have come up with in terms of satisfactory explanations for, is how is it that autoreactive cells and high-affinity antigen-reactive cells can simultaneously be selected for and against, because one might imagine that since these are autoreactive, they should be experiencing high avidity interactions because there's a boatload of self around all the time, and so this should be a pretty strong BCR signal, yet we're going to selectively eliminate that and selectively preserve these. I'd like to show you one little piece of new data that I think may be part of the answer to that puzzle. And it really began with our asking, is it possible that Bliss or BAF plays a role in this selection process analogous to how it plays a role in transitional cell differentiation and selection? And my postdoc, Radhika Genka, did this really lovely picture. And what she showed was that if you now stain, again, the germinal center with peanut agglutinin, and that's what's going to be all the red cells here, and I've taken the liberty of showing you the light and dark zone, and you look for bound Bliss, you see what you would imagine is the case in the follicular pool. Nearly all follicular B cells are coated with Bliss all the time. But interestingly enough, in the germinal center, there is very little Bliss bound to the B cells, and there's a filigree of Bliss in the light zone. To make a long story short, what we've been able to show is that the reason that there is Bliss in the light zone is because TFHs are producing it. They form a local source of Bliss in the light zone of the germinal center. And the reason most systemic Bliss is sequestered out of the germinal center is because these B cells have lost one of those two Bliss receptors. They've lost TACI, and so the relative concentration of receptors is much higher out here than it is in here. And so the only way a BR3 positive TACI negative B cell is going to get a Bliss signal is from light zone follicular helper T cells. And we have done a mixed chimer experiment to address whether that's important, and it turns out it is. So if you make a mixed chimera, which in fact is Bliss knockout bone marrow mixed with TCR beta knockout bone marrow, so that in the reconstituted chimeras, only the TFHs, the TFHs are all going to be Bliss bereft, whereas everything else should be fine. Systemic Bliss is fine because T cells don't make the Bliss that's the systemic levels. You let them reconstitute, you immunize. Among other data that I'm not going to show you for lack of time, what you notice is something really quite interesting, and that is that even though you get the low affinity IgM response to the immunizing hapten, the high affinity class switch response takes a tremendous hit. This is at day 14. And if you do sequence analysis of the V regions coming out of this response, what you notice is that contrary to, this is a statistical analysis of selective strength, contrary to the controls, which had wild-type T cells and have this relatively high degree of selective strength, there's far less selective strength directed towards higher affinity if the TFHs cannot make Bliss. So we're forwarding this model, which is that as cells enter a germinal center and undergo somatic hypermutation, they downregulate TACI. They become a fairly Bliss-bereft cell. The only way they're going to get Bliss is with a lengthy cognate interaction with T follicular helper, such that higher affinity clones will collect antigen and present it, because that's what B cells are doing for T cells in a GC, to the TFH. This cements germinal center character in both of these cells, but more importantly affords Bliss-mediated survival only for this cell. And although this cell needs Bliss a lot because it's in fact a high affinity BCR signal strength, it is not collecting the antigen and will not engage in this local presentation event that affords access to Bliss, and it will die. We are forwarding the notion that this actually allows an explanation for why you can simultaneously select high-affinity interactions for antigen and against high-affinity interactions for self within the same structure. So what I've basically presented so far is this idea that we have two major checkpoints, both of which Bliss and its receptors play a big role in, and that is at this immature or transitional stages where excess Bliss allows everything to get through because this is systemic, and at the germinal center level where we sort of drain the local environs of all Bliss, make it available in only one place, such that high affinity cells receiving a high BCR signal will only survive if they are also presenting the antigen. So can we exploit this? And I think that's what I want to spend the latter 15 minutes. How much have I got? 10? No. I used to check. I got 20 minutes. Oh, now we can really babble for hours. All right, or we can take a little time more time for questions. And so we posited some time ago that it might be possible to exploit this rather interesting relationship. That is, the more of this cytokine you have, the less stringent your selection is. And could this be exploited in one of any of three possible scenarios? And as I said, I want to show this to you more from the standpoint that, yes, we are beginning to achieve a knowledge of how lymphocytes are selected to the point where we can begin to fiddle with it. It may not be with this Bliss molecule, but that's the example I'm going to give you. But I'd like to throw it out that these are the things we ought to think about, really surgically screwing around with repertoires. And so I'm going to give you three examples. Well, I'm calling it the 3 R's. The idea of we might be able to resolve a variant selection. We might be able to revise selection and accommodate neo-self-antigens. This may be of greatest interest to this audience. And we might even be able to relax selection when we actually want things that normally are selected against, which might be of interest to folks that are trying to generate difficult to fathom vaccines. So I'd like to walk through examples of each of these three, which have, well, 2 of the 3 of which have been published. And I want to use as my example this little cartoon. What I'm showing you here is this selective process of all these different clones, good, bad, and indifferent, as they enter a Bliss-mediated selective checkpoint. And I've coded them to say that with the orange cells are autoreactive or polyreactive. The blue cells have too low a tonic BCR signal, so they're not going to survive. The green cells are good guys, and the gray one is a green one, but has an interesting specificity that we're going to call anti-Z. This will come up in my second example. And so normally what one sees is this truncation of specificities down to only those that we might want. And as most of you know, B ablation's become rather popular lately for a variety of reasons. And one of the drugs of choice is rituximab. It's an anti-CD20 antibody. And one thing to remember, however, is that when you do this, you create this situation. You go from this low Bliss environment to a fairly high Bliss environment because you ablated most of the users of this cytokine. And it occurred to us that it seems possible that when one does this, unless you keep doing it, if individuals reconstitute, you may have a period or a window in which this selection is no longer as stringent as it ought to be. And so the question that arose with us is, might it not be possible to, upon ablation, add or titrate a reagent in that can in fact ablate Bliss levels, fooling the system into thinking it is replete, at least in terms of Bliss sensing, and allow this selective process to occur normally. So I'm going to show you an example in a mouse, and you may see an example, I think, from Betty Diamond in the future where this is actually being done in SLE patients treated with Ritux to see if it holds up in people. But in mice, one thing you can use as a poor man's surrogate for appropriate selection is the proportion of B cells that bear lambda light chains as you move across the transitional to the follicular pools. And it turns out that during this selective step, lambda light chains are heavily selected against in the B cell pool. You go from about 10% to about 2% for reasons that probably have to do with the charges found in the V regions of lambda light chains and autoreactivity. And what we're going to do, therefore, is intentionally treat some mice with an anti-CD20 that we kindly got from Tom Tedder. And we're going to treat them also at the same time with a Bliss neutralizing antibody to achieve this idea of titered back Bliss. So let's see what happens. And the answer is right here. Sure enough, when you ablate with anti-CD20 only, Bliss levels go up, as you can see here at day 20, and day 40 they're starting to come back down. Whereas when you concomitantly treat with the anti-Bliss reagent, they stay reasonably low. You don't see this spike in coming back down. And anti-Bliss alone obviously takes out quite a bit of it. But what you do see here is quite interesting. With no titration or ablation of the Bliss at the same time as you ablate your B cells. Sure enough, this normal degree of counter-selection of lambda is lost until very late in the, is in fact far different than it usually is, and if it is in fact restored, these are different times after ablation, it is restored in the individuals where you can commonly treat it with Bliss, and so with anti-Bliss, very sorry. And so we think actually it's going to be possible to temporarily correct the system's perception of its need to fill its peripheral pool such that it is now we have an appropriate stringency at that selective checkpoint. As I say, there is a trial just beginning looking at exactly this combination and measuring exactly this point in humans, in SLE patients. So this idea of resolving selection may hold water, but the one I'd like to now talk to you about is this idea of actually revising. And here's old anti-Z. And you remember the Medawar experiment, which is you really can't cross a full allo-histocompatibility boundary without dire consequences. And because what's happening is, even though we normally select things in this individual's milieu, when you introduce an alloantigen, and I'm going to call that Z, you have not selected against that, and therefore you have a tremendous amount of anti-Z activity. However, it occurred to me and a few other people, some of my transplantation colleagues, namely Ali Naji and his laboratory, that it might be possible now to actually ablate and rebuild your B-cell pool around an alloantigen as long as you maintain an appropriate level of selective pressure at this point. And so the experiment's going to be, can we take an alloantigen, introduce it, but ablate and titer Bliss back so the system rebuilds itself around Z under normal normal selecting stringency. And so let's see. Here's the experiment. So what Ali did was he said, let's do an islet transplants, and what we'll do is we'll destroy the host islets at about day -6 with streptozotocin. We'll transplant C57 black into BAL-C. This is a full MHC mismatch. It's H2B into H2D. And then we'll treat with RAPA to take out the primary T-cell response. Believe me, if you don't do that, you will have graft rejection. But then once we've gotten over that hump, come in here, ablate the B cells totally, back here at day -20, but now titer down anti-Bliss until we achieve full B cell reconstitution. And here's the experiment. So the way you measure, as probably most of you know, the ablation of islets is you become, you have an awful lot of glucose in your blood after you, in fact, ablate. Here's the streptozotocin treatment. Their glucose levels go sky high. As soon as you put that C57 black graft in here, they come back down to normal. And what I want to call your attention to is that throughout that entire weaning period, which is up to about day 60, the grafts stay. That's in itself pretty amazing. Okay, just let me tell you, that in itself is pretty amazing. You would normally have gotten an allo response in here probably by about day 24 or day 30. Furthermore, it went on for basically indefinitely, but at this point the first grafts were removed, and I might point out we're doing nothing more to these mice. That we stopped the anti-Bliss at day 60, and then we add, and you see this glucose levels go sky high, then you give a second set of C57 grafts to this animal, and sure enough, with no further treatment, I find this very interesting, with no further treatment, you basically have full allo tolerance. I would add the third-party graft is rejected. So if you came in with H2K, you would reject that, albeit with somewhat slower kinetics. We could talk about mechanism a little bit if you want to, although this is published work. It's from the Naji laboratory. So I think this notion of revising really has some merit, or it might start telling us ways you could target things in order to achieve this idea of rebuilding a repertoire in a way that the normal homeostatic mechanisms are basically, what's the word I would like, that you've found a way to make the system believe that homeostatic mechanisms are acting normally and can rebuild around these or neo-self-antigens. Finally, I'd like to talk about the exact opposite, and that is maybe it's possible to relax selection to look for things you normally have a hard time finding. Now you might want to ask yourself, when on earth would you want an autoreactive B cell? Or maybe more accurately, when on earth would you want a B cell that you normally select against, that is normally the selective disadvantage? And I think one could argue there are times you could imagine that being of some value. Obviously, one time that might be is in neoplasia. I mean, if you want an antibody to a tumor, it is by definition an anti-self antibody. Similarly, there may be some pathogens that have mimicked us so closely that we just, in fact, can't get around it, and we're deleting the clones we want because it looks a lot like us. And finally, just maybe some cells never hit the appropriate tonic signaling level to be positively selected, and in fact, the pathogen has exploited that selective process. And so, to test this idea, we went right for the throat, and along with Pia Desinovic, who's a grad student in Nilla Hedestam's laboratory at the Karolinska, she got constructs of the GP140 HIV surface protein from Rich Wyatt, and asked the question, if we pre-treat animals with Bliss and then immunize with OMB, will we get a different quality response that contains antibodies we normally don't see? And are some of these more broadly neutralizing as you look across members of the same clade? So all A and B are showing you here is that the Bliss treatment worked. That is, at the last of the 10 days of Bliss treatment, the spleens weigh a lot more, and the reason they do is because they have a boatload more B cells. They have about twice as many B cells as they should have. But this drops back off once you've stopped the Bliss treatment. But I'll remind you that the immunizations occur right in this window where we have expanded the B cell pool and presumably have been letting things in that normally don't get allowed into those pre-immune pools, as I showed you way back at the beginning. And here are the data. This is a boost, the secondary boost, as well as a third and fourth boost with OMB. The point I'm making here is that this isn't different. The treated animals are not different than the PBS controls. But that's important. It's telling you the amount of antibody they made, which is exactly what this is. This is the amount of antibody. And the same was true for ELISpots as well. Really doesn't change, is not different between the treated and the control. You make about the same amount of total antibody. The difference was in the specificities you got. So in the first boost, what you saw was right away that viruses, and these are the non-immunizing viruses, these are heterologous HIVs, you have reactivity to viruses you didn't get when you immunized the PBS-treated controls, and this difference becomes greater and greater with successive boosts. So you can see in the challenge 3 times animals, the number of different viruses that we have detectable titers to is significantly greater than that which we got in the PBS controls. And so I'd like to leave you with this overall idea, as I'm exemplified by basically playing with Bliss, that it is in fact going to be possible as we move forward to consider manipulating repertoire. And we can manipulate it both to fix things we are screwing with ourselves, such as resolving selection when we've called it off for other reasons, such as in ablative regimes. We can revise it to our liking by exploiting the notion that, well, when Bliss is low, the system doesn't try to push and fill itself, and therefore we can rebuild repertoires around neo-self-antigens. And then finally, we might be able to expand repertoires in a way that will allow us to perceive at least specificities that we might like to call out, but that are normally lost at these normal selective checkpoints. And I'll stop there and entertain questions.
Thank you very much, Mike. That's really given us a lot of food for thought. I think certainly opened up some eyes with regards to some possibilities of how we can particularly apply this in the future. So sure, there's lots of questions. So hasn't really got to start, or maybe you've answered everybody.
It's not a B-cell line.
You think there's a stunned?
Well, perhaps. I could say one thing though. Who was the gentleman that spoke before me? William, are you here? Something I think is worth noticing, this idea that the immatures are the regs, or the B-regs, or that may be where B-regs come from. If you look back in this situation here, what you're also doing when you titer back bliss is you're sustaining the immatures as the majority of the pool for a much longer time, because you're not allowing this to reconstitute as quickly as you would if you let everything through. And there's evidence, twofold evidence from these experiments, that what's going on here is that these are going to be the major presenting B cell, and there's some thought that this tends to foster Treg formation. And because they are the majority of B cells, these transitional cells, that you will have a lengthier window in which to generate those sort of negative regulatory responses. And so it may not be so much building the pool around the NES-alpha antigen as much as sustaining an immature pool as the majority of your cells for a much longer window than you otherwise would.
Yeah, just a comment. So then how to potentially use this for, I guess, tolerization in drug treatment, I guess the sort of ablation regimen.
I'm not sure you'd want to do that. I mean, there could be...
Yeah, many normal scenarios not, but applicable maybe in some of the enzyme storage disorders where it's already used anyway. In some cases, to treat so these are patients like with
Really?
disease.
Oh, I didn't know that. Is that true? Do you ablate these folks?
Yes, well, because they're completely negative for the enzyme that's even as a therapeutic drug, and a high proportion of them basically, not having the enzyme is absolutely fatal over time. Giving the drug is rescues that, but the anti-drug response that develops in many of the patients, is also then potentially fatal if it persists and indeed ablates the effect of the drug. So then people come back in with taxane to the B cells.
It's probably after the horses left the barn, though, I would think,
yeah,
if it's done after the fact.
But, I think the challenge to overcome immunogenicity in that scenario is so great that I guess it does merit a plastic intervention potentially.
Well, certainly if they're already being ablated, I would argue, by the way, that Ritux, your big problem is exactly this. You do get rid of the offending cells, but then you have a selecting milieu, which is just basically letting everything through. So you're running a risk of autoreactivity as well as just rebuilding the pool exactly as it was before and facing the same problem. I would caution, however, that as far as we can tell, once cells are in a memory pool, I'm sure there are other homeostatic cytokines for a memory pool, but it's not this one. And as far as we can tell, the memory B cell pool is largely bliss independent, and therefore, if you've already got cells there, as you may have after you've already made a response, this would not work because you aren't going to fix the memory pool that way, unless you get rid of the memory pool and then rebuild the primary pool.
Yeah, I think in some cases the development of responses can be very well predicted in those patients when they're completely negative for the factor. So there is perhaps a rationale for actually pre-ablating.
No.
So I guess a slightly different angle on the same sort of question, but if we're looking at preventing immune responses in the first instance, would there be a way to, at the same time as we're treating with our therapeutic, be able to alter this kind of pathway, or at least the revision stage, so that when we're looking at an element maybe of receptor editing, that we can somehow fool the immune system into thinking that what we're giving them is self?
Well, as I said, I think if you're just dealing with a primary pool and not with pre-existing responses, that for B cells, I think that should be possible with this or modified approaches of this sort. The question you would have is the one I sort of recoiled at when is that not everybody is going to be a candidate for for B ablative therapy, and one must also worry, I think, about, like we did have to take out the entire primary T cell response with rapamycin in order to make all this work. But yeah, but yes, I think this is the arm. I mean, this is a lot better than generalized immunosuppression, perhaps. But this is still an arm and hammer approach. This is saying take everything out and rebuild it properly. But there may be targeted ways you could take parts out and then rebuild just that piece of it. That would be a little less, draconian as far as the approach is
I
concerned.
appreciate it may be a little bit too speculative at the minute to have an idea of what elements you might hit with that.
No, I don't.
So, Martin, in the context of the transplantation experiments you were talking about, your collaborators, have you also looked at other kind of similar experiments where, rather than looking at rapamycin, or inducing more of a tolerance approach rather than just kind of kicking in with a drug? Is that been done?
Well, the prediction. So, in terms of the T cell side of it?
Yes, exactly.
Well, no, not to my knowledge, but the implication based on the very late removal and putting back in of C57 into the valve is that they're fully T cell replete, so they're T cell tolerant too.
Yeah.
And why that is, that's why I raised this point with respect to Williams' and earlier remarks, that it may well be that sustaining a transitional pool of B cells as your major B cell component, because that's what it does. It's like 80% of your B cells for a much longer time, because you're rebuilding the repertoire but not filling that the follicular pool quickly. It may be that those are particularly good at generating either regulatory T cells or T cell or global T cell tolerance, because the implication is they're T cell tolerant too, because that's an untreated mouse.
Exactly. Yeah.
But it's had an H2G. The mechanism, I think, is something that needs some work.
Very interesting. Yes.
What are your thoughts on memory B cell populations which might not have been selected for in germinal center reactions, like say
Unswitched?
Right, yeah, so CD27 positive IBD unswitched.
Yeah, well.
B cells, and then in the context of that population, the role that Bliss might play if in fact.
There's not a lot on that. So I won't sort of touch the loaded question of whether or not those cells ever saw a germinal center, because that's a very difficult. I mean, because they do show mutations, which is rather interesting. On the other hand, AID can come up in a non-germinal center cell, so are they just an early exiter from a GC reaction? There's a mature pool, a memory pool, or not? But directly to your question, there's not a lot of data, as far as I know, in humans about the sensitivity of the unswitched memory pool to the ablation or neutralization of Bliss. But there is data in mouse, and we did this in collaboration with Marshall Omchick's lab. And the observation was that while switched memory, and this was done in a mouse model where we immunized within 60 days and treated with anti-Bliss and asked about the primary pool and the memory pool and memory responses. And the observation was that switched memory is clearly just insensitive at all. They could care less whether there's Bliss around or not. I mean, you ablated the entire primary pool, but that memory pool persisted and made normal responses. But the switched memory pool did take about a 40–50% hit, and so they may retain some degree of sensitivity. And before you ask me, no, we don't know what the receptors are on those things.
Thank you very much.
All right, thank you.